Welcome back to my channel Biology by Dr. Kumar Kurve. Today we are going to talk about a few details about embryology and gametic cells. To begin with, let's understand the detailed structure, let us say the ultrastructure, the microscopic structure of a human sperm or a mammalian sperm. First of all, it's an ultra microscopic cell, haploid cell, that's divisible into two main parts. There is a head and there is a tail. So human sperm, as I mentioned, would consist of a head and a tail. Now what is head? This is the head. The rest all is tail, which will be divisible into different parts that I would talk about. In the head, here is an acrosome. Acro means tip. Some comes from lysosome. So it's a lysosome present at the tip of the sperm, hence it's called as acrosome. This is a haploid nucleus. Haploid nucleus. Next, now the tail will be divisible into this. Actually, it's identified as the mid piece. Between mid piece and the head, there is a neck. Just above the neck, there is a part of centriole. This is called as proximal centriole. Now, passing through this midpiece and continuing further is an axial filament, or it's also called as axoneme. Axial because it defines the axis, passes through the center or central axis, and neme stands for a fiber. So a fiber-like thing that is passing right through the central axis. In the mid-piece, all around this axoneme are these mitochondria. Now, at the end of this tail, or let us say the midpiece, somewhere here would be the distal centriole. Then this is tail proper, which continues. See, this is around 5 microns, this will be around 50 microns. It's a very long tail. So this would end with This is continuation, and here is extension of the axoneme. So this is continuation of the tail proper, and here is the end piece. So mid piece and head piece. Further, mid piece would be a part of the tail. This is central part of the tail, and end piece again would be part of the tail. Each part has a specific function. If you talk about the acrosome, acrosome contains an enzyme, a sperm lysine you can call it. Or hyaluronidase. I'll write it here. This is meant for piercing the female gamete, the egg cell, oocyte. How does that happen? When this reaches the covering of the egg cell, the acrosomal cap, it breaks open and the sperm lysine or hyaluronidase is released out. This tries to penetrate, this tries to dissolve the covering of the egg cell and let the sperm enter. But of course, we must understand that a single sperm directly cannot penetrate to the oocyte and get in for fertilization. The first sperm comes in, releases the sperm lysine, a small dent is made, second sperm follows it, the dent is deeper, third sperm, like that a number of sperms reach there, and finally there is a sperm who releases the sperm lysine, makes a small hole in the covering of the egg cell and gets in for the purpose of fertilization. All earlier sperms, after releasing the sperm lysine, they are simply disintegrated. They are sacrificed, and there is one which is able to enter in. So, acrosome or acrosomal cap is meant for releasing an enzyme, that is sperm lysine, which would help in entering the female gamete or the egg cell. That is how it happens. The nucleus is the haploid nucleus, which is responsible for carrying the male set of chromosomes into the female gamete, so that the zygote has half the chromosomes from female, half the chromosomes from male. Then the centriole, proximal and distal centrioles, these are actually meant for defining the poles at the time of cell division. Cell division in the sense the mitotic divisions of the zygote, the fertilized egg cell. So these two are required for defining the poles for the astral spindle to be formed for the mitotic divisions. I am not going to the details of the division because you must have learned it elsewhere. Axoneme or axial filament is nothing but structurally similar to a typical cellular flagellum, the 9+2 kind of flagellum. That is the axoneme or the axial filament. The mitochondria are meant for generating energy. See, the thing is, sperm is released at one place and it has to reach another place for the purpose of fertilization. That is, let us say it's released into the vagina and it has to reach the fallopian tube for fertilization. So from vagina till fallopian tube, its movement is possible by spending quite an amount of energy, which is generated by the activity of mitochondria. So mitochondria are meant for generating energy that help in movement of the sperm. Fine, then the axoneme, the axial filament continues further. Yeah, this is rest of the tail and this is end piece as I mentioned. Now end piece is nothing but extension of the axial filament, the axoneme, similar function. The wriggling of the tail, fast movement of the tail would help in movement of the sperm, that helps it in shifting from the place of release at the time of ejaculation till the place of fertilization, that is from vagina till fallopian tube. This is what the sperm, the mammalian sperm looks like. Now let's try and understand the detailed structure of mammalian egg cell. So the human egg cell or the mammalian egg cell, it's better known as an oocyte. It's not called as an ovum. That's because the cell that we're talking about is actually an immature female gamete. It is yet to mature into an ovum. What you're seeing here, what you're going to see here, it's a secondary oocyte. In the developmental procedure, this oocyte has to undergo second meiotic division so as to give rise to the ovum. So it is not an ovum, it is an oocyte that we're talking about. It's a haploid cell. This is the haploid nucleus, around which this is a routine plasma membrane. There's one more covering around. This is called as a vitelline membrane. Around this vitelline membrane, there is one more layer. A thick jelly-like layer would be seen. Now, outside this, there is one single nuclear structure. Let's name the parts. This is the egg cell. Here is the haploid nucleus. This is the plasma membrane, around which is the vitelline membrane. Here, this jelly-like layer is zona pellucida, and here it's a polar body which is going to disintegrate in further course of time. That is, if at all there is fertilization. So this is the female gamete which is released out from the ovary for the purpose of fertilization. In addition to all this, around the whole thing, there would be these cells. Keep in mind, these are all diploid cells. Can be named as follicular cells. Cells are part of cumulus oophorus. Part of cumulus oophorus, or exactly if I have to label this layer, it's called as corona radiata. These are the follicular cells, diploid cells. Now it's a microlithical or almost an alithal egg. The amount of yolk contained is negligibly small. So what you see here is predominantly cytoplasm with a single haploid nucleus. That is what the mammalian egg cell looks like, or that is what the mammalian egg cell is seen after it is released out from the ovary into the coelomic cavity, into the body cavity. Let's then have a look at egg of a fish. Egg of fish. First of all, it's a macrolithical egg. It contains large amount of yolk. Moderately macrolithical egg, and further it is telolithical. That is, the egg yolk and cytoplasm, they are distributed in two parts. One part is completely yolk, that is called as the vegetal pole. The other part that contains cytoplasm and nucleus is called as the animal pole. These two parts are clearly distinguishable. Now, what does the egg look like? This is all yolk. This is all yolk. Around it, is a covering. Around yolk is the white line membrane. This will contain even fat globules in it. This is actually actual cell. You can almost call it to be the animal pole-like region. In this, here is where the embryo would be formed. The embryo would be formed in this region predominantly. So let's label the structures. This is all yolk. This is the embryo. This is the embryo. Covering the whole thing is a layer that's called as the chorionic layer or chorion. That's mainly the covering of the entire structure. Now here there is a small cavity which is called as perivitelline space or perivitelline cavity, which will have a significant role in the future process of development, embryonic development. This is what the fish egg looks like. As you can see, quite some part of this egg cell is occupied by yolk. Some region is the embryonic region where the animal pole will be there. This will be vegetal pole. The whole thing is covered over by a chorionic layer. White line membrane will be covering this central part. This is what the fish egg looks like. Now let's know the frog egg. This is also a macrolithic and telolithic egg. Large amount of yolk. It is also moderately macrolithic and yolk is located on one part of this cell. So animal pole and vegetal pole are clearly distinguishable. So what does it look like? This could be the cell, plasma membrane, around which there would be an additional layer. One more layer further around it, that's a thick jelly-like covering. Okay. There is a haploid nucleus. This part would be majorly the animal pole. All cytoplasm and nucleus will be contained in it. This is all yolk, so this is a vegetal pole. Now you can note, let's label the parts. This is the jelly-like layer. This is the vitellite membrane. Here is the plasma membrane. This is animal pole that contains cytoplasm and nucleus. This is vegetal pole. And here is a region that is between animal pole and vegetal pole. This is called as the gray crescent. This has an important role in the development of Blastula and Gastrula. You will note when we talk about that. So this is what the frog egg would be looking like, unfertilized egg. Note the animal pole and vegetal pole, they are clearly distinguishable. Animal pole contains cytoplasm and nucleus. Vegetal pole contains yolk. The whole thing is surrounded, in addition to these two coverings, by one more layer that's called as a jelly layer. This jelly layer would see to it that all the eggs, they stick together, they stay together, and they form a spawn. Cluster of those attached eggs, it's called as a spawn. Okay, which is possible because with jelly coverings that are present around the egg cell. That is the frog egg. Then let's talk about egg of a hen. What does it look like? First of all, here also it's a macrolithic and telolithic egg, but it is a largely macrolithic egg. Huge amount of yolk will be contained in the cell, and so the animal pole, the part of the cell that contains cytoplasm and nucleus, is only on one side of the egg cell. It forms almost a small disc, which is then called as a blastodisc. This is the only region which will contain cytoplasm and the nucleus. So blastodisc will be clearly distinguishable. That is actually the animal pole. Okay, let's understand the structural details. Suppose that this is the entire egg cell. This area would be distinguishable. Now what you see here, this is supposed to mainly contain the yolk and the embryonic cells, as the animal pole as well. There is an additional covering next to it. There. This region is marked as chalaza. Then this is the central region where embryonic development would be taking place. Now this is where the yolk would be found in concentric yellow and white circles around a flask-shaped region yellow and white circles will be there. Then here it would be the nucleus. This, as I said, is chalaza. Now, these are the yolk circles. Then, in this region, there is albumin, but this is a thick albumin, and this is thin albumin. This flask-shaped region, it's called as latebra. It again has an important role in the development of embryo. Now here there is one more layer, a relatively thin porous layer would be a calcareous layer. This is a hard shell. This is a calcareous outer membrane. Then there would be one more layer. This is the inner membrane. Here is an air space, air cavity. that helps in providing oxygen to the developing embryo, because this shell is almost impermeable. A hard shell it would be there. So calcareous shell is a characteristic of bird eggs. So this shell would be almost impermeable, and hence this provides the breathing space for the developing embryo. This is what the hen egg would be looking like. Let's then know about the cleavage process. What is cleavage? It is the mitotic division of the fertilized egg, that is zygote. Mitotic divisions of the zygote are called as cleavages. Now, what kind of cleavage would be applicable to which particular egg would depend upon how much yolk the egg cell contains. Whether it is alecythal, microlecithal, megalecithal, moderately megalecithal or largely megalecithal, that will decide as to what kind of cleavage would be there. Quite some part of this I have elaborated in the earlier session. So here I would focus mainly upon the structural details which you can observe under the microscope. Okay. First of all, cleavage in Amphioxus. Here it's a macrolithic egg cell. So the cleavages would be holoblastic. Holoblastic and of course equal to a good extent. Let's know why. Holo means entire, blast means fission, division. The division is complete, that means each zygotic cell or derived from the zygote, the cell would divide into two daughter cells distinctly. Two distinct daughter cells will be formed during every cleavage. That's why it's called as holoblastic cleavage. And the daughter cells thus formed would be almost of the same size and shape. Hence it is equal. So it is holoblastic and equal. How does the cleavage take place? The first cleavage is vertical. You can call it as meridional. Second cleavage is also meridional, but it is perpendicular to the first one. So how do we show that? Suppose if this is the zygotic cell, this is the diploid nucleus. The first cleavage would be in this manner. This is one daughter cell, this is another daughter cell. Second cleavage is perpendicular to the first one, so this is how it will go, resulting into 4 daughter cells. Third cleavage is horizontal, but it is slightly above the equatorial plane, slightly above the equatorial plane. So the cells thus formed would not be exactly of the same size and shape. Slightly above the equator. So we can identify these as micromeres and these as megameres. In general, these cells will be called as blastomeres. Why so? Mere means a part, blast means a fission. So part of the cell separated, formed because of the fission, blastomeres. And here micromeres because these are smaller, megameres because these are little larger. This is the kind of cleavage that would take place in Amphioxus. Further, these cells will continue to divide. Okay, so these cells will divide into relatively smaller cells, micromeres, and these will divide into relatively larger cells, that is megameres. Okay, mind well, the difference is there, but it's not so remarkable. We will know that when we actually have a look at further stages of development. Then we will talk about cleavage in frog. We have called it to be a macrolithical and telolecithal egg cell. So the cleavages are holoblastic but unequal. That is, complete daughter cells will be formed, but the daughter cells need not be of the same size and shape. Here what happens, suppose if this is the zygotic cell, as usual the first cleavage is meridional, second cleavage is also meridional but perpendicular to the first one, the third cleavage, the time would be these are now micromeres, these are megameres. Here the size difference between micromeres and megameres is clear, very clear. Distinct differentiation would be there because yolk amount is relatively large and most of the yolk is contained in the megameres and most of the cytoplasm is contained in micromeres. Let me tell you why there is a difference in the cell size. See, when the furrow starts developing, this is the furrow, the cleaving process. When it passes through the cell, it can clear the cytoplasm distinctly into two parts very easily. But when it is trying to divide the yolk, now yolk being a fatty material, it takes little longer for the yolk to be divided into two parts. So the furrow can easily pass through the animal pole but takes little longer to pass through the vegetal pole. And hence in the third cleavage, faster dividing furrow stays in the upper half and slower dividing cleavage or furrow remains in the second half, lower half. So these cells continue to divide little faster, these cells take little longer to divide. And so in future stages of development, these cells divide faster, become smaller and become larger in number. These cells divide slower, so remain larger but divide little slower. So that differentiation would become more conspicuous in further stages of development in the case of frog. So it is holoblastic and unequal kind of cleavage. Then let's talk about hen. What kind of cleavage it would happen. First of all, let's keep in mind this is a discoidal egg cell. That is, huge amount of yolk is contained, so embryonic material, the animal pole, is displaced into a small disc-like region, which we have called as a blastodisc. So whatever cleavage is applicable only to blastodisc. Rest of the cell, it's nothing but yolk. This is the blastodisc, suppose. So cleavage would remain only in this peripheral region. First cleavage, second cleavage, and so on. Now what happens here? In some stages of development, we come across a condition, a situation wherein part of the cell is facing the cytoplasm, part of the cell is facing the yolk. The part facing the yolk doesn't have a membrane there, so it's almost like an open cell. Part of the cell which is facing the cytoplasm, it does have a membrane. So these become the open cells at some stage. Open cells they will become. And these open cells would in due course of time give rise to something called as a periblast. Some of them would form the covering, they would become marginal periblast cells, and some would grow into the embryo, they would become subgerminal periblast or subgerminal periblast. 5 to 6 layers of these cells will be found in due course of time, subgerminal and when marginal for that matter. Okay. So this two cleavages are seen, third cleavage, fourth cleavage, and then there will be formation of masses of cells here. The periblast cells which are formed here, they would become the marginal periblast cells, and those which would grow into the embryo, they would become the subgerminal periblast cells. This is the kind of cleavage which is seen in hen, or let's say birds. Then let's have a look at the blastula stage in the embryo formation. That is, we have seen the division up to the 8 blastomere stage. That is up to 3 divisions, 3 cleavages we have seen. The cleavage process continues, and then we come across a sphere of cells enclosing a huge cavity which is called as a blastula. To begin with, let's talk about blastula of an Amphioxus. Amphioxus. Here it's called as a coeloblastula. A large cavity. Cel stands for a cavity. So blastula with a large cavity inside. Now if you recollect, we have seen that the micromeres and megameres are there, but the differentiation is not so sharp that you can clearly identify the difference. Some minute size difference would be there. So this is what the coeloblastula and in Amphioxus would be seen. Now what you are seeing here is the vertical section of the blastula. So this is a single layer of cells enclosing a cavity. This cavity is called as blastocele, and the layer of cells here, it's called as blastoderm. That is what you see as a blastula in Amphioxus. Then let's talk about blastula in frog. Here, as I've already mentioned, the difference between micromeres and megameres is remarkably observed. So if suppose this is the entire sphere that we're talking about, these are the micromeres, and these are the megameres or macromeres. So the cavity, if you observe, this is the blastocele cavity of the blastula, which is covered almost on 3 sides by micromeres, and the 4th side, the floor, is made up of megameres. Now mind well, this is also a coeloblastula. A large cavity is there. So this is what you will see as blastula stage in a frog. Then let's talk about blastula in hen, that's a bird. Here, if you recollect, we've seen that, so this is the entire sphere. The stage is going to be a discoblastula, because here we have seen there to be a blastodisc which develops into an embryo. So in this whole sphere, rest all is the yolk, and only in this region would be seen the cells. So this is going to be the blastoderm. This is all yolk, and this is the blastocele. So most of the sphere is nothing but yolk. Only a small disc-like area would contribute towards formation of a blastula. Okay, and so there is a small, relatively smaller cavity, a disc-like cavity covered over by blastoderm. That is the cells derived by mitotic divisions of the zygote. So that is blastula of a hen. Next embryonic stage is gastrula. Now, as we are talking about only vertebrates, this gastrula can also be called as a 3 germ layer stage. That is the stage wherein all the 3 germ layers, ectoderm, mesoderm, endoderm, all 3 germ layers are seen to be developed, or they are seen to be present. To begin with, we will talk about gastrula formation, that is gastrulation in Amphioxus. Here, I'm not going to talk about the process of gastrulation. Let us focus more upon the structure, the detailed structure of gastrula. First of all, the gastrula would be forming by the process of involution. That is, whatever sphere we had seen as a blastula, it would start kind of invaginating, and then it will be giving rise to the gastrula. So the gastrula that you see at an early stage, it could be something like this. There's one layer in the periphery. There's a cavity inside. Now the cells continue to multiply, and then you come across a stage wherein the cells have divided and have resulted into formation of one more layer of cells in this. The cavity is now called as archenteron. The outer layer would stay as ectoderm, inner layer would be endoderm, and in due course of time, one more layer will be developed in between, that would become mesoderm. So finally, the gastrula that we see is this is the outermost layer. As I said, it's ectoderm. Second layer this is the third layer, innermost layer, that is endoderm, and here is going to be the mesodermal layer. Now here a small opening is formed, that would become the blastopore, which will give rise to the gut, and later on that would develop into an alimentary tract. So this is what you see as the gastrula in Amphioxus. Let's know about gastrula in a frog. As we have seen, here the blastula stage would clearly indicate it's a coeloblastula, of course. 3 sides covered by micromeres and the 4th side covered by megameres. Okay. Now here the process would involve invagination of these cells from one particular point. Earlier we have named that as the grey crescent, from where the cells would start invaginating. That is, these cells, they divide and start forming another layer here. The second layer, outermost layer. This is the outermost layer. This is going to develop into ectoderm. The second layer is going to develop into endoderm. And as you can see, one more cavity is appearing there. This is the archenteron. Archenteron. Gradually this would grow and it will always replace the blastocyst. This would grow here. And it would almost replace the blastocyst. This is the kind of development which will happen in frog. Now what happens in the mature blastula? Here this becomes the yolk plug. Yolk laden cells are emerging out from here. Rest of the cells, they are giving rise to ectoderm in the periphery, endoderm in the at the base, and mesodermal layer would start appearing here. Okay. In the yolk plug, there is a dorsal lip. Ventral lip will be formed below. This is the kind of gastrula which you see in the case of frog. Okay. In the case of birds, we have seen hen as one of the examples. Now in hen, as we have already seen, the process of development would be restricted to only a particular region, that is blastodisc as we have called it. In the blastodisc, the cells are proliferating, the cells are multiplying, and then there is a stage wherein this is one layer of cells, this is all yolk. The same layer of cells, now let's stick to only this region now. If you recollect, I said that some cells are open cells. One side of the cell is in contact with the yolk. These cells would be giving us to one layer here that would become hypoblast. This would become epiblast. Let's keep in mind the entire embryo would be developed from epiblast cells. Hypoblast would not have any role in the formation of embryo. The epiblast cells will continue to multiply, continue to divide. Here it is all hypoblast. The cells, they will multiply. from both the sides that will happen. The process is now delamination. A layer is being detached, and this would gradually give rise to endoderm. This is going to be ectoderm. Now here this tiny depression which you are seeing, it's called as primitive streak, which should have a significant role in the formation of future stages of embryo. Now some of the cells, they are detached from here, and these cells, they start forming one more layer, which then becomes mesoderm. Okay, so in the final stages, there would be one more layer here. This would become the endoderm. One more layer of cells will start arranging here. That will become mesoderm. This is the kind of stage you would see as gastrula in a hen or a bird. I'm sure you must be enjoying these lectures, so don't forget to like them, subscribe to them, and share among your friends and family. If you have any queries, suggestions, doubts, don't forget to post them. Thank you.